nanomaterials, the procedure we have developed in controls. It's Anders Bavn who's speaking, and Lars Mikael Skjolding is also here from DTU, and we would share the presentation. So I'll give you a bit of background, and Lars will come in later and tell you a bit more about the technical details. Just a second. So the settings is that we have a webinar that will not take more than 75 minutes. The presentations will be recorded and will be available on YouTube afterwards. And we will share the slides with all the registered delegates. We will ask you all to be on mute, so the sound quality is better. But of course, during the questions and answers session, there will be some unmuting. And the questions can be asked via the chat function that is in the GoToMeeting panel. Our program for today is to give an introduction to the controls project and to our approach for algal testing. Then we will present our our LEVITATT toxicity test system, and then there'll be the questions and answers session. And the aim is to give you an overview of the challenges that are when we want to test nanomaterials in algal toxicity testings, and then to show our approach for how to increase the reliability and the availability of algal toxicity testing using our LEVITATT approach. Controls is a EU project, and it's called Physiologically Anchored Tools for Realistic Nanomaterial Hazard Assessment. And as you can see in the long text here, we have a long aim, but we highlighted in red what is important to notice in this context, and that is that we will provide innovative and effective sets of laboratory techniques to reliably predict potential human and environmental hazards that can result from exposure to nanomaterials. So we use the abbreviation ENM in some of our slides for engineered nanomaterials. Control's solution to do this is that we are developing and establishing a battery of innovative next generation testing tools so that we can predict the effects of exposures to nanomaterials in the environment. So we will deliver different innovative methods. We will also work on computational tools, and we will make sure that we characterize nanomaterials before, during, and after testing, so that we can both be better in explaining what happens in the test, but also contribute to model development. We focus in the environmental part very much on the regulatory relevance and reliability of the test, so that we ensure that they can actually be used for safety assessment of nanomaterials. This is because there's an urgent need for tests that are actually relevant and reliable when we want to identify, rank, and classify nanomaterials for their environmental hazards. And as illustrated, it's often the OECD guidelines that governs this. But there's also a need to have relevant and reliable methods when we want to establish safe levels in the environment, the so-called predicted no effect concentrations. We depart from the notion that modified guideline tests already have the status of regulatory relevance, and that means that we know that they are relevant for the decisions that have to be made. It doesn't necessarily imply that they are environmentally relevant, but they are relevant for answering the questions at hand. However, it's also a realization that the reliability of these tests may be seriously challenged when we want to transfer them to nanomaterials. The regulatory reliability of Ecotox tests is usually taken care of by this function that we have indicated in red here, or that says that the effect is a function of the concentration, where we try to keep everything else stable or defined. So this little mathematical symbol means that given all the other things are kept kept constant. In plain terms, it means that a concentration response curve can be established since the effect is only caused by the concentration of a chemical. However, the picture below shows what we have experienced many times. This is from a PhD thesis of Sarah Sørensen here from our department, where she, after many years of Ecotox testing, said that she thinks it looks like this, the concentration response curves, that everything is a bit shaky on the axes, and everything happens under time, and therefore it can be a bit difficult to actually figure out what's going on in the tests. This means that the reliability is challenged, and that the reproducibility of test results can be very difficult to work with, since we actually don't know which are the influencing factors of this. Basically, everything could pitch in. This is most likely because there's a number of processes occurring for nanomaterials that we're not used to seeing when we are dealing with dissolved chemicals. Here's a range of them. All of them can contribute to these axes that we saw on the other picture. In patrols, we have focused on a few guideline tests that we said here we think we can make a difference, and we can improve them and make them better. And that's the algae test and the Daphne test. And it will be the algae test that we are focused on in this webinar. So why is reliability an issue for nanomaterial testing in algae test? Well, here's some electron micrographs of different particles being tested with algae. The algae are these more like a banana shape that are here. And we see a behavior of particles that is very far from what we would see if we tested a dissolved chemical. There is ongoing processes, and it raises a lot of different questions about what actually happens biologically. Some would claim that when we do the algae testing, we actually only contribute by raising more questions than actual answers. This is not very good for the reliability of the testing. It's interesting and fun for scientists, but it is a big problem for the regulatory use of the tests. So basically, we could say there's a number of processes that pitch in and makes this situation quite bad. And that in the circle here, you could say here's a lot of scientific issues to try to figure out all these equations. But that's not the angle that we have taken in this part of the work in patrols. We also focus on that. But for our test development, we have realized, and this is not genius, but it's just a fact, that all of these processes are strongly influenced by the testing conditions. Algae tests have a reputation for being one of the most difficult, or if not the most difficult, of the standardized tests to actually get to behave properly. And we have a long-standing experience in working with the algae test, and we have also contributed to both guidelines and standards in this area, and realized that it's really a lot to do with testing conditions if you want to make a good test. So the algae, they are simple organisms in one way, but they demand many things to be kept stable if you want to have a good reproducibility. We would like to have stable pH, often ensured by a good CO2 influx. They also need the CO2, of course, for creating biomass. We need to have defined light in good light fields within certain ranges, because their growth rates and so on are very dependent on the light. And temperature control is very crucial and often goes hand in hand with, if you want to increase the light, we also get a higher temperature, and therefore we decrease the light, but then the algae do not grow optimally. We can get problems with shading, for example, in nanomaterial testing. We also need to have some shaking or other way to keep the algae in suspension, and probably also to keep the nanoparticles in suspension. Finally, there's like a very practical issue about testing volume. So if you want to obtain some of the other things like defined light or or good and uniform temperature. It's not a good idea to make too large flasks, but on the other hand, we need to take out samples to characterize things if we are testing nanomaterials, and we also need to be able to actually count the algae in safe and reproducible ways. So we need a volume that's small but not too small, but big enough to do characterization before, during, and after, and we need to do biomass determination, of course. This could be standard biomass like cell counting and so on, but it can also be things we're working on in patrols, other types of endpoints where we are focusing a lot on algae pigment content. So the patrols approach to making better algae tests is actually to propose a defined testing setup, and in that way we aim to increase the reliability of the tests. Our design principles for doing this is that we sort of need to minimize the degrees of freedom that is given by the OECD guideline. If you read it a bit more carefully, you'll see that they give, of course, a number of good advices and suggestions in a guideline. That's what a guideline is. Whereas the ISO standard, being a standard, is much more strict and says you have to do like this and like this. And we have chosen the ISO standard as our template for defining what would be the optimal test conditions. We have a design criteria that said it has to be small scale, but not mini scale. We want to increase the testing capacity and availability, but we can't go all the way to micro scale. We think if we need to have samples enough to document that the last point can be fulfilled, that is that the ISO validity criteria are also in place. So these things have led to our development of the LEVITATT testing platform that I will now hand over to Lars, and he will give you an introduction to that.
Yeah, my name is Lars Skjolding, and I will present to you the LEVITATT, which is an LED vertical illumination table for algae toxicity testing. Basically, the LEVITATT was developed to increase the reliability and availability, as Anders just mentioned, and it was also developed in order to be able to test more difficult substances such as volatile substances or shading or colored substances. This is done through the use of versatile test containers that can basically be changed in order to meet the needs that you need for the given substance. We have a complete description of the setup and a video of how we are performing the experiments that have been submitted to the Journal of Visualized Experiments. The reference is seen below. The initial point of departure could be the test volume, as Anders also mentioned. It's very important to have a sufficient test volume, especially when you're testing nanomaterials, to be able to characterize and quantify your behavior of nanoparticles during your test setup. We are using 20 milliliter scintillation vials with 4 milliliters of media inside where the algae is also situated. This enables a steady and nice CO2 flux throughout the test, which also ensures a stable pH. Also, when we're using small vials, we can increase the number of replicates per concentrations, and the small size also ensures that we have sufficient biomass in order to determine the cell concentration. Here is the LEVITATT, and it's situated inside a regular thermo cupboard. The dimensions are approximately 45 centimeters by 45 centimeters and approximately 30 centimeters in height. This allows it to be situated in also smaller incubators, so it increases the availability and also the space required to actually perform this type of test. Regularly, you would use Erlenmeyer flask or larger setups that would require much more space than presented with the LEVITATT. Basically, this test setup allows for 45 vials to be used for testing each time with each LEVITATT. And To exemplify it, it could be that you're testing one LJ test with one compound at 5 given concentrations, and then with 6 controls, for instance. Here it's seen inside the thermocouple and just from slightly above. So you can actually see that the LED source is located in the middle of each casing, and around the LED source is a mounting of clamps on the outside casing. This allows for a homogeneous light field throughout the testing period, and it also, due to the incorporation of the LED sources, it allows for a minimal heat generation. The metal clamps allows for different versatile test containers to be used. You can use different glass vials and just clip them into the metal clamps. Here the levitate is seen from the side. Basically, we have the LED sources sandwiched in between 2 plastic sheets, and then it's mounted on an orbital shaker. Mounting the whole system on an orbital shaker can be done with either sticky mats. So basically, it's just a mat that is situated between the orbital shaker and the upper part of the plastic sheet. And basically, it allows to have the whole container just sitting on the orbital shaker without moving too much. Furthermore, you can install a dimmer, so you can adjust the light field. Basically, you can adjust it with this setup anywhere from 0 to about 160 micro-Einstein per square meter per second. Basically, this corresponds approximately to 4,000 to 8,000 lux. This is well within the ISO guidelines, and it also allows for taking into account the difficult substances such as colored substances or nanomaterials that can cause some different types of shading, for instance. It has been advised that you can increase the light intensity. This is also possible with the levitate without adding further heat generation. Also, the light is situated below the samples, so there is no shading of the samples by other samples or from the lid itself on the vials. So the advantages of the levitate is that it has an adequate sample volume, as Anders also mentioned, so we are able to do destructive sampling. For instance, characterization of nanomaterials, for instance, for size or for concentration. Also estimation of LJ pigments, if there are changes in the pigments due to colored substances, or they are shaded by intershading or by localized shading of nanomaterials, for instance. It also allows for quantification of biomass after 24, 48, and 72 hours, for instance. The compact size increases the testing capacity, but also the availability in terms of lab space required to actually perform the experiment. The well-defined and adjustable light field allows for testing at many different light intensities, but also for testing of more difficult substances such as colored substances or nanomaterials that might cause shading. Using the LED source, we have a very nice temperature control in terms of that we don't heat up the samples and we don't generate much heat in general due to the low energy required to actually run the LED sources. There is a sufficient CO2 flux that allows for a constant pH and LJ growth. This is also promoted by placing it on the orbital shaker, so we have shaking ongoing throughout the experiment. The versatile test containers could also be closed vials, where you use an enrichment of the headspace for testing of volatile substances. Of course, all of these claims are somewhat justified by us doing a lot of tests with the levitate. We have tested 2 different reference substances and 3 different nanomaterials from the JRC repository. The results are shown in this table. From what we can see here is that the coefficient of variation is approximately, or at least below the coefficient of variation that was carried out by a round-robin study by 9 laboratories using the ISO standard for algae toxicity test. But generally, we can also see that even for the nanomaterials, we have a decent coefficient of variation. that somewhat complies with the ISO standard. In the patrols, we have initiated a round-robin test. It will be used to determine the interlaboratory reproducibility of the LJ test if we follow the protocols that have been developed in the patrols and also using the LEVITATT. So the aim of the interlaboratory test is twofold. One is to actually test the setup as it is, as the LEVITATT, but also in combination with the patrols SOP. Right now we have approximately 8 laboratories participating, but there is of course option for more laboratories to join in. If you are interested, you can contact me in this email. The round-robin test for the patrols entails that you will be testing 2 test substances, one that is an organic test compound and the other is an inorganic nanomaterial. This is basically done to have a control that allows us to test a regular chemical in the LEVITATT test setup, and then we will also try to test a nanomaterial. An SOP and equipment will be provided by DTU if you choose to be a part of the round-robin testing. So here is a brief summary. Basically, the LEVITATT have been developed in the patrols to increase the reliability and availability of standard algae toxicity test of nanomaterials. Here is briefly summarized all of the advantages of using the LEVITATT, and basically what these different advantages do is that it will allow us to actually have compliance with the current OECD and ISO validation criteria. This will also be validated by our round-robin test. And last, I would like to thank Patrols for funding. Patrols is also able, you will also be able to follow the Patrols project on social media, for instance, LinkedIn or Twitter, or you can subscribe to our yearly newsletter. And we would like to thank Horizon 2020 for funding under the grant agreement number.
Thanks very much, Lars. We can open to questions now after that extremely straightforward and understandable presentation. Thank you very much. I'm going to stop the recording now so that the Q&As are more offline.